Good morning, students. Today we will discuss about DNA structure. DNA, which stands for deoxyribonucleic acid, is a very important biomolecule found in most of the living organisms. Before going into our topic, let us recollect the experiments carried out by Mendel. He just carried out several experiments by some sativa, and he just explained the mechanism of inheritance. According to him, all the characters are controlled by particulate factors. Now we are calling these factors as genes. Then, what is the chemical composition of a gene? Gene is basically made up of DNA, deoxyribonucleic acid. Then what is the basic function of a gene? The genetic information of all living organisms except the RNA viruses like plant viruses is stored in DNA. That means DNA will store the information and it will transmit the information from parents to the progeny. Both DNA and RNA are large molecules and may occur as either single or double-stranded states. The nucleic acids were first isolated by F. Miescher in 1869 from the nuclei of WBC of pus cells, and he called as nuclein. Actually, the term nucleic acid was coined by Altman. DNA is an acid because of its phosphate groups, hence the term nucleic acid. It contains a sugar, deoxy-D-ribose, and therefore is called as deoxyribonucleic acid. Then what are the types of nucleic acids here? There are two types of nucleic acids, deoxyribonucleic acid, which serves as a cellular database by storing an immense amount of information regarding all possible polypeptides a cell can make. The other nucleic acid is ribonucleic acid, which occurs in several different forms like messenger RNA, ribosomal RNA, transfer RNA, and is needed to convert DNA information into polypeptide sequence. As you know very well that the flow of information will be always from DNA to RNA by transcription process and from mRNA to protein by translation process. In some viruses, RNA serves as the primary database with no DNA involvement. You can give the examples of so many plant viruses and retroviruses where DNA is absent, RNA is acting as genetic material. Certain RNAs have catalytic ability similar to that of a protein enzymes, and these are called as ribozymes. And here, nucleic acids are built from subunits called nucleotides. Here, nucleic acids are the polynucleotides. They are having many monomeric units. Each monomeric unit is called as nucleotide. Let us see what is the structure of a nucleotide. Each nucleotide includes three components. A ring-shaped molecule, we are calling it as a nitrogenous base. The bases are of two types, purines and pyrimidanes. And the second one, it is a 5-carbon sugar, which is a pentose. Here the sugar is ribose in case of RNA. The sugar is deoxyribose in case of DNA. And the other component of a nucleotide is a phosphate group. This nucleotide may be having one or more phosphate groups. See here, I can see the basic structure of a nucleotide here. This is a pentose sugar in the form of furanose ring structure. 5 carbon atoms are there. The pentose sugar is actually ribose in case of RNA. That's why we are calling as ribonucleic acid. The sugar is deoxy without oxidation, deoxyribose in case of deoxyribonucleic acid. Now this sugar is linked to a nitrogenous base, maybe either purine or pyrimidine, by N-glycosidic bond, and at the same time this sugar is also linked to a phosphate by means of an ester bond. So this is a basic structure of a nucleotide here. So let us see one by one the bases. First, bases are of two types, purines and pyrimidines. Pyrimidines contain a six-membered pyrimidine ring, which is similar to benzene ring, except that it contains nitrogen in the place of carbon at position 1 and 3. Purines are double ring compounds, which consist of a five-membered imidazole ring joined to a pyrimidine ring at positions 4 and 5. In DNA, there are four different bases: adenine, guanine, these two are the purines; cytosine and thymine, these two are the pyrimidines. RNA also contains four different bases. Three of these are same as in DNA: adenine, guanine, and cytosine. RNA contains uracil instead of thymine. So as I earlier mentioned that a base is joined to a sugar by means of N-glycosidic bond. So if you are strictly speaking, at N1 of pyrimidine and N9 of purines, they are linked to first carbon. Here I'm using prime, is 1 prime. It just indicates the position of carbon in sugar. So the bases are linked to first carbon of the pentose sugar by means of a covalent bond. The covalent bond here is N-glycosidic bond. And the phosphate group is linked to fifth carbon by means of an ester bond. The N-glycosyl bond is formed by removal of water, a hydroxyl group from the pentose, and a hydrogen from the base. DNA and RNA can also be considered as containing two kinds of bases. So based on the functional groups, we can also categorize the groups as amino bases and keto bases. Amino bases, basically they are having an amino group, whereas keto bases, they are having a keto group as the main functional groups. Amino bases are cytosine and adenine, and keto bases are thymine, guanine. In case of RNA, it is uracil. We just see the structure of purines and pyrimidines. As I mentioned here, purines, they are having two rings. So one is pyrimidine ring and other one an imidazole ring. So here this is the numbering of purines and pyrimidines. And here I can see the pyrimidine structure just mimics the benzene ring, except that at first and third position, instead of carbon, nitrogen is there. And here we have to remember the numbering also. In case of purines, ninth, the nitrogen which is present at ninth position is linked to first carbon of the sugar. In case of purines, the ninth nitrogen atom is linked to first carbon of sugar by means of N-glycosidic bond. You have to remember this point. Whereas in case of pyrimidines, first nitrogen is linked to first carbon of sugar by means of N-glycosidic bond. I already mentioned that this glycosidic bond is formed by the removal of water molecule. So hydrogen atom is contributed by the base, and hydroxyl group is contributed by sugar. Now just see the structure here. Adenine, 6-amino purine. The basic structure of guanine is 2-amino 6-oxy purine. Coming to pyrimidines, in case of DNA, it is thymine, 2,4-oxy pyrimidine, and cytosine, 2-oxy 4-amino pyrimidine. In case of RNA, instead of thymine, uracil is there. If you observe the structure of uracil and thymine, the thymine structure is 2,4-oxy 5-methyl pyrimidine, whereas in case of uracil, it is 2,4-oxy pyrimidine. Only thing, methyl group is absent in uracil. Thiamine, how can we name this thiamine? 2,4-oxy 5-methyl pyrimidine, and other one uracil. So it is just thymine without methyl group. Thymine without methyl group is uracil. And nucleotides, it is having a base and sugar. So here the sugar found in nucleic acids are pentose. It's a 5-carbon atom. It's a 5-carbon sugar, and the sugar is ribose, D-ribose, dextroribose. Ribose found in ribonucleic acid. It's a normal sugar with 1 oxygen atom attached to each carbon atom. Deoxyribose. Deoxy means without oxidation. The term itself indicates that 1 oxygen atom is absent. Deoxyribose found in deoxyribonucleic acid, DNA. It is a modified sugar. That means it is lacking 1 oxygen atom without oxidation, deoxy, at second carbon. So what is the sugar in deoxyribose? It is a pentose sugar, ribose without oxidation. It's a ribose, D-ribose without oxidation, it's second carbon. So we are calling that sugar as deoxyribose or 2'-deoxyribose. Just see the structure here. It's a condensed furanose ring structure, cyclic structure here, D-ribose. So this ribose, it's having hydroxyl group at second position, whereas deoxyribose, it is lacking oxidation, 1 oxygen at second carbon. Here we are giving the numbers as prime, 1', 2', 3', 4', 5'. It is only to differentiate the number of carbon from the bases. In bases, in purines and pyrimidines, we are just using 1, 2, 3, 4, 5 like that. Whereas in case of sugars, we are using prime. This prime itself differentiates, it indicates that is the position of carbon in sugar. For example, I'm saying 3'. That means you should know that is the third carbon of ribose sugar. Now, coming to phosphate group, I mentioned that nucleic acid, because phosphate, this group is derived from the phosphoric acid. So that's why DNA is having acidic nature. We are calling it as acid. Phosphoric acid has 3 reactive hydroxyl groups, of which 2 are involved in forming the sugar phosphate backbone of DNA. A phosphate moiety binds to the 5' carbon of one and the 3' carbon of other neighboring pentose molecule of DNA to form the phosphodiester bond. You just remember that a phosphodiester bond is formed between 2 nucleotides. How those 2 nucleotides are linked here? By means of 2 ester bonds, phosphodiester bonds. This phosphate is linked to 1 sugar at 5th carbon. This phosphate is linked to carbon at 3rd position. So that's why we are calling this as 3', 5' phosphodiester bond. Nucleotides typically have 1, 2, or 3 phosphate groups and are named as monophosphate, diphosphate, or triphosphate accordingly. You just see here a nitrogenous base and a sugar. Here it is having a base and a sugar, pentose sugar, base and sugar. So sugar is linked to base by means of N-glycosidic bond. Base with sugar is called as nucleoside. Now to the nucleoside, if phosphate moiety is added, phosphate group is added by means of phosphoester bond, then it is called as nucleotide. Nucleoside plus phosphate group It's nothing but nucleotide. And these nucleotides are linked with each other by means of 3' 5' phosphodiester bond. It's a dinucleotide. Like this, if many nucleotides are linked together, it is forming a polynucleotide. Just see here the basic structure of nucleotide here. So actually this is a ribose sugar because it's having a hydroxyl group at second position. Now this is the first carbon and this is the base. So here this is a purine. This is an adenine. Now this is the ninth nitrogen. Ninth nitrogen of purine is linked to first carbon of the sugar by means of N-glycosidic bond. Here you can see the removal of one water molecule. So hydrogen is coming from base and hydroxyl group is coming from the sugar. And if this sugar is linked to a base, that is called as nucleoside. Now to this nucleoside, if a phosphate is added, it will become a nucleotide. Then how this phosphate, this is a derivative of phosphoric acid, then how this phosphate is added, how this phosphate is linked to the sugar. This phosphate is linked to sugar at fifth carbon by means of an ester bond. Here you can see the ester bond. Here this is the glycosidic bond. So these are the covalent bonds. They are very strong bonds. So if oxygen is absent here at this place at second position, it will become a deoxyribose sugar. You just see here. Now this is the sugar with the base. Sugar with base, it will become a nucleoside. Sugar base with one phosphate. It is nothing but nucleoside monophosphate because only one phosphate group is there. That phosphate is linked to sugar by means of an ester bond. So to this nucleoside, if two phosphates are attached, that is nothing but nucleoside diphosphate. If three phosphates are attached, it will become nucleoside triphosphate. If you know the structure of these purines and pyrimidines sugars, then it will be very easy to write the structure of all nucleotides. Only thing you have to remember the position at which the bases are linked to sugar and the phosphate is linked to sugar. You can see here. Now, a polynucleotide has a free phosphate group at the 5' end of the sugar, and this is called the 5' end. Similarly, the sugar also has a free 3-hydroxyl group at the other end of the polynucleotide, which is called the 3' end. The backbone of a polynucleotide chain consists of pentose sugar and phosphate groups, whereas the nitrogenous bases project out of this backbone. Just see, this is the polynucleotide chain. Poly means many. Here many nucleotides are linked. I mentioned that nucleotides are linked by means of phosphodiester bond. Two ester bonds are there. This is one ester bond. This is another ester bond. Now this is a nucleotide. See here, this is a nucleotide, or we can say nucleoside monophosphate because only one phosphate I have taken here. Now, to the first carbon, a base is attached. To the fifth carbon, phosphate is attached. And this nucleotide is linked to another nucleotide by means of 3', 5' phosphodiester bond, because this phosphate is forming an ester bond with the third carbon of this sugar. At the same time, the same phosphate is also forming an ester bond with the fifth carbon of another neighboring sugar. So this is 3', 5' phosphodiester bond. So like this, if all the nucleotides are connected by phosphodiester bonds, the last nucleotide definitely it is having a free 3-hydroxyl group. So if you are just taking this polynucleotide chain, so here this is a first nucleotide. So this is having a phosphate group, free phosphate group at 5' end. And if you just continue the same nucleotide at the end, so definitely that should be having a free hydroxyl group at 3' end. Now, so here 5' end with free phosphate group, and this is the 3' end with the free hydroxyl group. So this is called as 5' phosphate sugar linked by 3' 5' phosphodiester bond. So like this, if you go on like this at the last, this is 5', you are starting with 5', that means this fifth carbon is having free phosphate. If you go on like this, definitely that last nucleotide should have free hydroxyl group. Now, so can you pronounce these words? These are the nucleotides actually. Yes, you are right. Deoxyriboadenosine triphosphate, deoxyribose adenosine diphosphate, deoxyribose adenosine monophosphate. What are these? They are nothing but nucleotides. A combination of base and a sugar is called a nucleoside, For example, a base adenine is added to the sugar deoxyribose. Sugar and base. The sugar is deoxyribose and the base is adenine. Definitely you are getting a nucleoside. What is that nucleoside here? Deoxyribonucleoside. As it is having adenine base, we can name that nucleoside as deoxyadenosine. Deoxyadenosine. Here, what does this A indicates? Adenosine. Prefix D is there, deoxyadenosine. That means this is a nucleoside. DA, deoxyadenosine, deoxyadenosine. This is a nucleoside. That means deoxyribose sugar is linked to adenine. Now, here it is having monophosphate, diphosphate, and triphosphate. If this deoxyadenosine is linked to one phosphate, that is deoxyadenosine monophosphate. If that adenosine is linked to two phosphates, it will become DADP. If it is linked to three phosphates, it is nothing but deoxyribose adenosine triphosphate. In that way, we can just name the nucleotides of DNA. Just see here, maybe there may be deoxyadenosine mono, di, or triphosphates. Like that, you can just give the name as DGTP, DGDP, DGAM, GMP, like that. Deoxyadenosine monophosphate, deoxyadenosine diphosphate if it is having two phosphates, deoxyadenosine triphosphate if it is having three phosphate groups. So this is the basic structure of DATP. Definitely you will be knowing the structure here. Just see the sugar first. This is not having any oxidation at second carbon, so the sugar should be deoxyribose. And here it is linked to the base. What is the base here? Adenine. Okay, only adenine and ribose if you are talking, then it will become a nucleoside. So what is the name of this particular nucleoside here? Adenosine, sorry, deoxyadenosine, because it is lacking one oxidation here. So we should call this particular nucleoside as deoxyadenosine. Now, this deoxyadenosine is linked to one phosphate. For example, only one phosphate is there. That will become deoxyadenosine monophosphate. If it is linked to two phosphates, it will become deoxyadenosine diphosphate. If it is linked to three phosphate, it will become deoxyadenosine triphosphate. So like that, you can just write the structure very easily if you know the basic structures of purines, pyrimidines, bases, and the linkage at which position exactly they are linked to the sugar. Just see the nucleotides in DNA, how we can just give the names. Nucleotides, for example, base is adenine. That means you should know that the base is adenine and the sugar is deoxyribose sugar. Then what we are calling that particular nucleoside? That is a deoxyribonucleoside. If the base is adenine, that will become deoxyadenosine. If it is a nucleotide, deoxyadenosine monophosphate. If it is one phosphate, DATP, DADP, like that. If the base is cytosine, the name of the nucleoside here it is deoxycytidine. And coming to the nucleotides, DCMP, DCDP, DCTP, cytidine monophosphate, deoxycytidine diphosphate, deoxycytidine triphosphate, like that. If the base is guanine, it is deoxyguanosine. You can give the name deoxyguanosine monophosphate, deoxyguanosine triphosphate, like that. Thymine. Thymine is present only in DNA, you know. In RNA, thymine is absent. So deoxythymidine, it is a nucleoside. Coming to the nucleotide, deoxythymidine monophosphate, deoxythymidine diphosphate, like that. And here, if you are just talking about the nucleotides in RNA, so what is the simple difference here? In RNA, instead of deoxyribosugar, ribosugar is there. That means we should not use the prefix deoxy. Very simple. We are not using the prefix deoxy. Okay. And one more thing, instead of thiamine, uracil is there. For example, in case of RNA, the base is adenine, and the sugar is obviously it's a ribosugar. Adenine plus ribosugar. It is a ribonucleoside. What is the name here? Adenosine. So there is no prefix deoxy, adenosine. Then what are the ribonucleotides here? Adenosine monophosphate. You are not using prefix D. Adenosine diphosphate, adenosine triphosphate. Okay, like that you can give the names of nucleosides and nucleotides in case of RNA. Cytidine, guanosine, and here there is no thymine, uracil is there. And the nucleoside name is uridine, uridine monophosphate, uridine diphosphate, uridine triphosphate. If you know the basic structures of purines, pyrimidines, sugar and phosphate group and their linkages, then you can write the structures. You can easily write the structure of adenosine, AMP, ADP, ATP. Okay, so you can just make it as an assignment. You have to write the structures of all these nucleotides in case of DNA and in case of RNA. Now just by seeing, you can just give the name. This is an assignment for you. Just can you say the names of these nucleosides here? See here, this is having deoxyribosugar. Okay, the base is cytosine and only one phosphate group is there. So we can call this nucleotide as deoxyribocytidine monophosphate. So like this, you can give the names of all these nucleoside monophosphates. So what I mentioned here, only one phosphate is there. So all these are the nucleoside monophosphates. Now this is simply UMP. This is DTMP. So like this is TAMP. This is DTMP. So like this, you can give the names. And coming to the structure of DNA. So till now we have seen the composition, the chemical composition of DNA. So let us see the structure of DNA here. DNA known to contain purine, pyrimidine bases, deoxyribose and phosphate. But how they are arranged? In 1947, Chargaff published data showing that percentage of adenine, thymine, cytosine and guanine showed certain regularities. Percentage of bases varies from organism to organism. And the percentage of adenine is equal to percentage of thymine. And percentage of cytosine is equal to percentage of guanine. This is called Chargaff's rule. What did it mean? In 1952, X-ray pictures of DNA taken by Rosalind Franklin in Wilkins' lab in London showed some kind of helix. In 1953, Watson and Crick published a model built from Franklin data. It is nothing but a double helix structure. This double helix structure suggested that Chargaff's rule was due to base pairing. What is base pairing here? Which type of base pairing here? Adenine pairs with thymine and cytosine pairs with guanine. So that's why we are getting the percentage of adenine is equal to percentage of thymine. Percentage of cytosine is equal to percentage of guanine. Now, two chains of DNA face in opposite directions called anti-parallel. It is defined by which way 3' and 5' sides of sugar molecules are facing. In linear molecule, one strand has free 3' end, whereas the other strand is called as complementary strand. It has 5' end. So here you can see this is anti-parallel direction. DNA is having two polynucleotide chains, So this is one nucleotide, this is another nucleotide, and the direction of these two nucleotides will be opposite. Opposite means anti-parallel. What do you mean by anti-parallel here? So now this polynucleotide is having 5' end. The other nucleotide is called as complementary strand. Now this is having free 3' end. If this strand is having 5' free end, the complementary strand should have 3' hydroxyl free end. So this is called as anti-parallel direction. You just see the structure here. So this is one nucleotide, polynucleotide. This is another polynucleotide chain because DNA is having two polynucleotide chains. And all the nucleotides are linked by means of phosphodiester bond. You know very well how this phosphodiester bond is formed. If you observe this polynucleotide chain, so this is having free phosphate group at 5' end, and the last one should have 3 free hydroxyl group, and its complementary strand should be in anti-parallel direction. That means if here phosphate group is free, definitely it should have 3 hydroxyl, free 3 hydroxyl. Here it is having free 3 hydroxyl. Definitely its complementary strand should have free 5' phosphate. And these two polynucleotide chains are joined by means of base pairing. Okay, so base pairing. Adenine pairs with thymine by means of 2 hydrogen bonds, guanine with cytosine by means of 3 hydrogen bonds. So let us see salient features of DNA double helix here. It consists of 2 polynucleotide chains where the sugar and phosphate group from the backbone and the nitrogenous bases project inside the helix. The 2 polynucleotide chains have anti-parallel polarity. That is, if one strand has 5' to 3' polarity, the other strand has 3' to 5' polarity. The bases on the opposite strands are connected through hydrogen bonds forming base pairs. Adenine always forms 2 hydrogen bonds with thymine from the opposite strand and vice versa. Guanine forms 3 hydrogen bonds with cytosine from the opposite strand and vice versa. Just you have seen the picture. Therefore, a purine always pairs with a pyrimidine on the other strand, giving rise to a uniform distance between the 2 strands of the helix. The 2 strands coil in right-handed fashion. So here, these 2 strands, they coil in right-handed fashion. Each turn of the helix is 3.4 nanometers or 34 angstrom units, consisting of 10 nucleotides. These nucleotides are at a distance of 0.34 nanometers or 3.4 angstrom units. The helix is stable because of the base pairs that stack over one another and hydrogen bonds that hold the bases together. Coming to Chargaff's rule, as I mentioned earlier, so he just is a biochemist, discovered that the number of nitrogenous bases in the DNA was present in equal quantities. The amount of adenine is equal to thymine, whereas the amount of cytosine is equal to guanine. See here, A is equal to thymine, C is equal to guanine. Why? Because adenine always pairs with thymine by means of 2 hydrogen bonds. That's why always the percentage of A should be equal to the percentage of T. And cytosine pairs with guanine by means of 3 hydrogen bonds. Definitely, the percentage of cytosine should be equal to the percentage of guanine. In other words, the DNA of any cell from any organism should have a 1:1 ratio of purines and pyrimidine bases. It is very clear that a purine is pairing with a pyrimidine. So obviously, total number of purines should be equal to total number of pyrimidines. Then the ratio will become 1:1 purine to pyrimidine basis. See, so how these base pairing, we are calling it as complementary base pairing. So how the bases are joined here. Guanine pairs with cytosine by means of 3 hydrogen bonds, just see here. And thymine pairs with adenine by means of 2 hydrogen bonds. Or we can say purine pairs with pyrimidine. And here adenine purine is pairing with the thymine pyrimidine. Guanine purine pairs with cytosine pyrimidine. So this is called as complementary base pairing. Now these are the nucleotides. They are linked by means of phosphodiester bond. So this strand is having a free phosphate group at 5' carbon, and the same strand is having 3 hydroxyl group at this end. This is another strand. This is a complementary strand which is in anti-parallel direction. So it is having free 3 hydroxyl end, and here this is having free phosphate group at 5th carbon. You see the structure, double helical structure, 5' to 3', one strand, and other strand is 3' to 5', anti-parallel direction. You can see a major groove and you can see a minor groove here. And sugar phosphate backbone, and the bases are joined by means of hydrogen bonds. So this is about the basic structure of DNA. So just I want to give some questions to you and just go through it and just come with the answers. So let us discuss this activity part in our next class. Just note down all these questions and you can think about it. So let us discuss about this in our next class. Okay. Thank you.